I'm Gabrielle H, a master's student at the University of Saskatchewan, working under the supervision of Dr. Ferrari. The research project I'll be discussing examines whether the concept of one size fits all applies to tadpole body size and antipredator behavior. When we think of a fear response in animals, one of the first concepts that comes to mind is the fight or flight response. More specifically, this is the neurological response to threatening stimuli or situations which triggers the sympathetic nervous system to increase in activity. This results in various physiologic changes which help increase an animal's survival against impending threats, such as predators. While predation threats trigger a single neurological response, animals have a wide variety of antipredator responses behaviorally, which can be broadly broken into three categories: fight, flight, or freezing, which we'll consider to be hiding. For example, three-spot damselfish have been found to freeze when there is a mild predation risk, flee, exhibited as a rapid dive and zigzag pattern when there is a high predation risk, and fight when the individuals are large in size. A single species can exhibit all three of these anti-predator responses because it's a behaviorally plastic trait, behavior that can be modified by various stimuli. So an individual must perform a rapid evaluation and choose which anti-predator response to perform in a given scenario. This is known as threat-sensitive predator avoidance, where a prey evaluates the level of risk, considers the current conditions and stimuli, and then selects the most appropriate anti-predator response as well as the magnitude of this behavior. The factors that can influence this decision include predator properties such as size, number, and proximity of predators, environmental factors like temperature and habitat, and the individual's own traits such as body size, morphology, and life stage. This concept of body size is a factor we're going to focus on, and how it influences threat-sensitive predator avoidance, both the size of the prey species and the predator. For the prey species, opposing sizes have advantages using opposite responses. Smaller individuals are less conspicuous when motionless, making freezing an advantageous option. Conversely, larger individuals have more muscle mass, making them more likely to escape or fight off a predator, so fleeing or fighting is a better choice. When evaluating the size of a predator, a small individual should elicit a weaker antipredator response in the prey because it's less likely they will mount a successful attack. A large predator should elicit a stronger antipredator response because it's more likely an attack will be successful. We wanted to examine this principle in aquatic species, where the decision-making process during predation events is primarily mediated by alarm cues, chemical compounds released from damaged epidermis tissue. They are interpreted by other individuals of the same species as a danger signal. Therefore, it influences a number of behavioral, morphological, and life history adaptations, as well as an overt and immediate antipredator response of either fighting, fleeing, or freezing. Our test species for our study was woodfrog tadpoles, which generally don't fight. So we're going to ignore that behavioral response from now on. In the alarm cue system, there are both donors and receivers of the alarm cues, allowing us to examine how body size of both these groups affects the antipredator response. We wanted to determine if the body size of alarm cue receivers influences the decision to freeze or flee in response to alarm cue exposure, and if the body size of alarm cue donors influences the decision of the recipients to freeze or flee in response to alarm cues. The idea being that this has the potential to act as a proxy for predator size. If a larger tadpole is injured, it was likely attacked by a larger predator. We began by sorting our tadpoles into three treatment groups based on size: small, medium, and large. We then exposed each of these groups to either alarm cues made from small, medium, or large tadpoles, or a water control. We monitored their antipredator response, which we quantified as their change in activity from before versus after cue injection. This is along the y-axis in our graphs. A decrease in activity indicates freezing behavior, the most common antipredator response in tadpoles, and an increase in activity could indicate a fleeing antipredator response. There's a lot on this graph, so we'll begin by breaking it down. First, all tadpoles did undergo a freezing antipredator response, regardless of body size, as they all decreased in activity compared to our water control. However, the magnitude of this response did vary depending on size. If we focus on the body size of the receivers, overall, small tadpoles responded to alarm cues with a stronger freezing response, or a greater decrease in activity. Larger tadpoles responded with a weaker freezing response, or a smaller decrease in activity, and medium tadpoles had an intermediate response, as we would expect. There is a gradient to this antipredator behavior, as small and large recipients are statistically different. However, medium recipients are not statistically different from either of them. Next, we can examine the body size of alarm cue donors. Here, tadpoles had a weaker antipredator response when exposed to cues from small tadpoles, and a stronger response, or a greater decrease in activity, when exposed to cues from large tadpoles. This supports our theory that injury to a larger tadpole indicates a higher predation risk, as it was likely attacked by a large predator. However, unlike the receiver size, the size of the donors doesn't follow a gradient, and instead has a threshold effect. Here, alarm cues from medium tadpoles were not statistically different from small tadpole alarm cues. But both small and medium alarm cues triggered a statistically smaller antipredator response than large tadpole alarm cues. Given that we didn't control for concentration, the most intuitive explanation for this variation in response to alarm cues is that tadpoles are interpreting the concentration of the cues. Larger tadpoles should release larger quantities of alarm cues than smaller tadpoles, which can then be interpreted as a greater risk. However, another possible explanation can't be ruled out. The chemical composition of alarm cues could change as the tadpoles grow or mature. Previous research shows that aquatic species have a complex chemosensory system, which is a bit more of an abstract concept. It is easy to understand how prey species evaluate predation risk visually, like the three-spot damselfish, who determines predator size, proximity, and body position based on visual evaluation of the predator. However, chemical evaluation can be just as informative. For example, fathead minnows can determine the proximity of predators from their odors, which is based on concentration, but they can also determine predator size and their number or density based on the odors emitted, independent of concentration. In itself, it is impressive how animals utilize threat-sensitive predator avoidance, a decision-making process to select a response to a single stimulus, a predator, a concept supported by this present study. Furthermore, our findings have the potential to add to the current body of evidence that there is a complex chemical system underlying how many aquatic organisms make decisions on predation risk. Further research is required to understand if alarm cues are interpreted solely on concentration or whether they follow a similar pattern of complexity as the olfactory sensory system, and to determine if alarm cues are either modified by the donor or interpreted by the receiver with a level of sophistication that surpasses our current understanding. And with that, I'd just like to thank my supervisor, Dr. Ferrari, as well as the whole lab crew. Thanks for listening, and let me know if you have any questions.